Good afternoon to each and everyone. I am Miley Ligardi, the next presenter. Today, I am going to talk about the different types of gene therapy. The first type of gene therapy is the somatic gene therapy. It involves the manipulation of genes in any cells of the body except sperm and egg cells that will be helpful to the patient but not inherited to the patient's descendants. So, somatic cell gene therapy involves the placement of human genes into a living person's somatic cells, a cell that do not produce the egg and sperm that in turn produce the next generation. Somatic cell gene therapy would aim to cure a disease only in the patient, not in the patient's descendants. Example is lung cells and blood cells. So, ang effects of gene therapy will not be passed on to the patient's children. Somatic cell gene therapy would aim to cure a disease only in the patient, not in the patient's descendants, in which you attempt to repair a defective gene or group of genes in a child or adult. The second type of gene therapy is the germline gene therapy. It involves the genetic modification of reproductive cells such as sperm and egg cells that will pass the change onto the next generation. So, ang germline gene therapy is when DNA is transferred into the cells that reproduce cells, eggs, or sperm in the body. This type of therapy allows for the correction of disease-causing gene variants that are certain to be passed down from generation to generation. Example ani is embryo and sperm. So, ang effects of gene therapy will be passed onto the patient's children and subsequent generation, in which you change the embryo to correct some expected or diagnosed genetic defect. So, the changed gene or the added gene can be transferred from one generation to the next. Have you heard about stem cells in the news? If yes, perhaps you wonder if they might help you or your loved one with a serious disease. You may wonder how they are being used to treat disease and why they are being discussed. So, ang stem cells are mother cells that have the potential to become any type of cell in the body under the right conditions in the body or a laboratory. Stem cells divide to self-renew or multiply. They can become cells of the blood, heart, bones, skin, muscles, and brain, among others. So, why is there such an interest in stem cells? Researchers and doctors hope stem cell studies can help to, number one, increase understanding of how disease occur. So, by watching stem cells mature into cells in bones, heart, muscles, nerves, and other organs and tissue, researchers may better understand how disease and conditions develop. Number two is to generate healthy cells to replace diseased cells or regenerative medicine. Ang stem cells can be guided into becoming specific cells that can be used in people to regenerate and repair tissues that have been damaged or affected by disease. So, people who might benefit from stem cell therapies include those with spinal cord injuries, type 1 diabetes, Parkinson's disease, Alzheimer's disease, and osteoarthritis. Ang stem cells may have the potential to be grown to become new tissue for use in transplant and regenerative medicine. So, researchers continue to advance the knowledge on stem cells and their application in transplant and regenerative medicine. The third one is to test new drugs for safety and effectiveness. So, before using investigational drugs in people, researchers can use some types of stem cells to test the drugs for safety and quality. New areas of study include the effectiveness of using human stem cells that have been programmed into tissue-specific cells to test new drugs. For the testing of new drugs to be accurate, the cell must be programmed to acquire properties of the type of cell targeted by the drug. Techniques to program cells into specific cells are under study. So, for instance, a nerve cell could be generated to test a new drug for a nerve cell, nerve disease. Tests could show whether the new drug had any effect on the cells and whether the cells were harmed. As for the ethical issues involved in gene therapy, there are many topics that are still in question, such as how can good and bad uses of gene therapy be distinguished. So, gene therapy does have risks and limitations. The viruses and other agents used to deliver the good genes can affect more than the cells for which they are intended. So, if a gene is added to DNA, it could be put in the wrong place, which could potentially cause cancer or other damage. So, ang current research is evaluating the safety of gene therapy. Future studies will test whether it is an effective treatment option. Several studies have already shown that this approach can have very serious health risks, such as toxicity, inflammation, and cancer. Will the high cost of gene therapy make it available only to the wealthy? So, gene therapy is very promising. It can be the treatment of the future for diseases like cancer and AIDS and hepatitis, and even diseases like hypertension and diseases and diabetes and others. We don't know how much effective can it be in the future, but many speculations suggest that it will develop into being effective in treating all the disease. However, other forms of treatment are also developing for every type of disease. For example, sa AIDS. Ang AIDS daw kuno is now curable, but the cost of the treatment is too high. Should people be allowed to use gene therapy to enhance human traits such as height, intelligence, or athletic ability? So, these are all questions waiting to be answered if gene therapy becomes more developed, researched, and clinically trialed. So, that's all for my report. The next topic will be the climate change, will be presented by Ms. Compayan.